Welcome to Food Safety 5, twice monthly reports from the Food Safety Magazine editorial desk to keep you abreast of top news stories and current events impacting the food safety sector. First, a recent study investigating biofilms in water hoses used in a meat processing facility revealed ubiquitous microbial colonization, reaffirming concerns about hoses as reservoirs for pathogens that could lead to cross-contamination in food production environments. For the study, hoses were sampled from 5 different locations throughout the facility at 3 different times across an 8-month period, and were replaced after each sampling event. The hoses, which were connected to a drinking water quality supply, were used for cleaning and sanitation of the equipment and facility, as well as supplying water for food production. A regular cleaning schedule for the hoses was not in place. Biofilms were detected in 14 out of 15 water hoses, which were made of thermoplastic elastomer and polyester, and most of which were used daily. The researchers identified and characterized diverse bacterial and fungal populations, including opportunistic pathogens and spoilage-associated bacteria, inhabiting the inner surfaces of the hoses. Despite uniformity in hose material, water source, and ambient temperature, microbial communities varied significantly between sampling points. Only one of the hoses was used on a weekly basis as opposed to daily, and that hose exhibited distinct microbial profiles compared to those used more frequently. Variations in water pressure and potential desiccation may have contributed to these differences. Interestingly, the presence of biofilms did not correlate with usage frequency or sampling time point, suggesting that other operational factors may influence biofilm development. Given the lack of a cleaning schedule for the hoses and the observed microbial diversity, the study underscores the need for routine monitoring and maintenance of water delivery systems. Next, a recent sampling and testing assignment conducted by Pennsylvania State University researchers demonstrated a low overall prevalence of Campylobacter on retail chicken meat. However, recovered C. jejuni strains did not match known poultry-associated genotypes, suggesting the need for sensitive detection methods and expanded genomic surveillance. Samples included 209 skinless, boneless chicken breasts collected from 7 grocery stores in State College, Pennsylvania, between October 2023 and July 2024. The researchers found a low overall presence of Campylobacter in just 1.9% of samples. Enrichments of 15, or 7.2%, of chicken breast samples produced visible colonies. Whole genome sequencing confirmed 4 isolates as C. jejuni, none of which matched common poultry-associated genotypes, indicating that existing databases may not fully reflect the diversity of Campylobacter in poultry environments. The findings underscore the importance of sensitive detection methods and expanded genomic surveillance to better characterize Campylobacter contamination and inform risk assessments. Moving on to Listeria news, another recent study investigating L. monocytogenes in South Korea's beef supply chain revealed the presence of the pathogen exclusively in retail product samples. A total of 445 samples were collected between April 2023 and October 2024 from both cattle farms and retail markets. L. monocytogenes was isolated solely from retail beef in 22 of 240 samples, or 9.2%, with no detections at the farm level. Genetic analyses revealed the coexistence of hypervirulent and stress-adapted L. monocytogenes subtypes in retail beef, reinforcing the need for continuous genomic surveillance. Additionally, the absence of L. monocytogenes in farm samples limits traceability during foodborne illness outbreak events, especially at abattoirs and processing facilities, which are critical control points for L. monocytogenes contamination, highlighting the importance of monitoring these establishments. Overall, the findings underscore the importance of enhanced hygiene practices post-harvest and robust food safety interventions across the meat production continuum. Lastly, we have 2 short news bites. The Cornell College of Agriculture and Life Sciences Food Safety Laboratory has developed the Salmonella Reservoirs Wiki, a web resource for global food safety professionals to rapidly access information about a given Salmonella reservoir. The Salmonella Reservoirs Wiki provides relevant information on more than 100 different Salmonella reservoirs and will be updated as more information emerges. Finally, the European Food Safety Authority conducted a literature review on the release of micro and nanoplastics from food contact materials into food and water. The experts concluded that, while there is clear evidence of microplastic release from food contact materials, the actual quantities are likely lower than many studies suggest. Additionally, studies on nanoplastics are severely lacking, and there are widespread methodological shortcomings throughout currently available studies. The current evidence does not support reliable exposure estimates. For more information on any of the stories mentioned in this video, be sure to check out the Food Safety Magazine coverage linked in the description. Check back twice monthly on Tuesdays for the latest Food Safety 5 episodes, available at food-safety.com or on our YouTube channel.